hear us now. So the Center for Bioethics, like I was saying, has a new Grand Round series. You can find out more about that on our website. Founded in 2001, the IU Center for Bioethics is a gathering place for discussion, education, research, and collaboration in bioethics among faculty and researchers across campus and across the state. And today it is my pleasure to introduce an accomplished scholar who has made significant contributions to this field of bioethics. Dr. In-Soo Hyun is director of the Center of Life Sciences and Public Learning at the prestigious Boston Museum of Science and is a member of the Harvard Medical School's Center for Bioethics. Dr. Hyun's work has focused on ethical considerations within the realm of stem cell research. Since 2005, he's been heavily involved in the International Society for Stem Cell Research. Notably, he was instrumental in drafting that society's international research guidelines and has twice served as the chair of the society's ethics committee. His intellectual interests extend beyond stem cell research, though, encompassing additionally emerging technologies in the life sciences and new and innovative strategies for community engagement in bioengineering. Dr. Hyun is also newly appointed member of NextTrack, a federal advisory committee that provides recommendations to the NIH director and a platform for thoughtful discussion of scientific, safety, and ethical issues associated with emerging biotechnologies. Dr. Hyun began his academic journey earning his bachelor's and master's in philosophy with honors in ethics and society from Stanford University, and he went on to complete his PhD in philosophy at Brown University. His expertise and insight have been sought on a national scale, and he's been interviewed frequently on National Public Radio and has served on national commissions for the Institutes of Medicine and National Academy of Sciences in Washington, D.C. Further, he is a regular bioethics contributor to esteemed journals like Nature, Science, and Cell, among many others. Today, we're privileged to have Dr. In-Soo Hyun share his wealth of knowledge and experiences with us, shedding light on fundamental ethics issues on moral status and personhood raised by new uses of stem cells in biomedical research. Please join me in welcoming Dr. Hyun. Thank you.
Well, good afternoon. I'm going to use this mic instead. Can people hear me okay? Okay, great. Well, thank you so much for this generous invitation to give the 2023 Olin Lecture. I want to thank the Department of Pharmacology and Toxicology and the Center for Bioethics for hosting me. And it's been a wonderful visit so far. And yeah, let me minimize this a little bit so I can see. All right, so I'm going to draw on some professional experiences, not only with the International Society for Stem Cell Research, but a lot of the close collaborations I've been doing over the years with leading scientists in the stem cell space. I have just a few disclosures to make. Okay, oops, okay. So I'm on the Bioethics Advisory Council at Bayer, and I'm a paid consultant for that. And I'm a non-paid member of the Scientific Advisory Board for Rumi Scientific, which is the company that aspires to use organoids and embryo models for drug screening. Okay. So the old debates. Back in the day of what I call the stem cell wars of the George W. Bush administration, this is really the time at which there was quite a bit of discussion about federal funding for human embryonic stem cell research and all these state initiatives that started popping up about finding alternative sources of funding outside of the NIH, for example, in California. During that very politically and ethically rife time, there was a lot of debate, but it mainly focused on the moral status of the human embryo that was derived from an IVF clinic from which you would get human pluripotent stem cells. At that time, there was only one source, which was the embryo, and there was no distinction between embryonic stem cells and pluripotent stem cells. They were basically the same thing. A lot of discussion around the moral status of these embryos, which otherwise would have been thrown away, and some discussion about some research that was starting where you were transferring human embryonic stem cell derived cells, human cells, into animals to create what are called human-animal chimeras, people who are trying to do that type of work. So the early debates, the predominant debates had to do with moral status issues, emerging personhood issues on entities that were being explored and used in the laboratory. And that characterized the debate for a very long time. So when I entered into the stem cell ethics field, this is kind of right at the beginning of, or right in the middle of this very politically rife time. And one of the things that happened reasonably soon after I arrived in the scene was the advent of human-induced pluripotent stem cells, which first was done in the mouse model in 2006 by Shinya Yamanaka and his team at Kyoto University, and then later was proven to be effective in the human case just about a year after that. But this seemed to be quite a boon for the stem cell field, because many people thought now we can finally get past the ethical issues and concentrate on the science. What happened here was they were then able to take any kind of somatic cell from a patient, from a healthy volunteer, reprogram them in the lab using viruses to overexpress genes that are very active in the stem cell state, and create essentially embryonic-like pluripotent stem cells. So this is the first time you actually had non-embryonic pluripotent stem cells. There was kind of a distinction between pluripotent, they could be induced or it could be embryonic. And when this happened, people would say to me, as a stem cell ethics person I was kind of sorting out at that time, Dr. Hyun, your work is pretty much done in the field, because now we have an ethics-free way of doing everything that we were hoping embryonic stem cells would do for both basic research and for clinical translation and therapies. The debates are over, we're done, we're in the clear. And I thought, just hold that thought, hold on for a minute. There was a big celebration at the Bush White House. In fact, at that time, when the mouse research was announced, so this is before they even went to human, George W. Bush had vetoed for the second time a bill that passed both houses of Congress. Can you imagine a bipartisan bill that passed both houses of Congress? And this happened twice. Went to the president's desk and he vetoed it both times. The second time, he said, there's exciting research coming out of Japan that shows that you could turn skin cells into stem cells. Therefore, there's no need to do embryonic stem cell research anymore. I'm even more convinced that due to our strict stance on preventing federal funding for use of human embryonic stem cells, scientists found a responsible, ethical way to move the research forward. So that was announced in the Associated Press when he signed this. He didn't mention that actually it was just shown in mouse right now. We haven't done it in human yet, but he made that announcement. And so I was, when he said that, I was on my way to an International Society for Stem Cell Research meeting in Australia. It was a long flight. I have time to think about that. When I arrived, I told Shinya Yamanaka, who's the person right to my right. This is an old picture from 2007, 2006 actually. I said, you know what George Bush just said to the Associated Press? He said, due to his strict policies, he motivated people like you in Japan to find an ethically appropriate way to move the research forward. And he said, I didn't care about the White House policy when I did the research. He was actually inspired by a research project done at the Harvard Stem Cell Institute using private money, because they couldn't use NIH money for this at the time, where researchers fused a human skin cell called a fibroblast with a human embryonic stem cell. And this enormous fused cell in its entirety behaved like an embryonic stem cell. So Shinya thought there must be something about the skin cell, the stem cell, that teaches the skin cell to do what it's doing. Instead of fusing the cells together, can we overexpress genes that are already present in the skin cell to teach it to become like an embryonic stem cell? So that's how he kind of got the idea of doing the project that he did, where he used retroviruses to overexpress the genes in this fibroblast. So it wasn't because of Bush's policy, but it was actually a natural curiosity spurred on by privately funded research at the Harvard Stem Cell Institute that he got the inspiration for this project. And he said, in fact, you actually have to do both together. You can't just stop human embryonic stem cell research and understanding of how human embryonic stem cells behave and just concentrate on the induced pluripotent stem cell types, because you have to constantly compare the two. If you're going to make an artificial version of an embryonic stem cell, you better really know what the real thing can do and compare the two. So it's kind of like a call for having both proceed together, both areas of stem cell research. So I told them, we should probably write this up and explain this. And so in the picture, we have Rudolf Janich, Conrad Hochlinger and Shinya and I, so on my flight back from Australia, I wrote up a little commentary. This was the eventual title of it, if you can see. No, sorry, I'm trying to get rid of this little box. There we go. And so I wrote it up, and then they added to it, and we published it in Cell Stem Cell. It ended up being one of the most cited papers of 2007, where we said you have to do both together. There are scientific reasons and there are ethical safety reasons why you have to assess both. So I'll get back to this idea of validation of a new technology using kind of the real thing. I'll get back to that idea of validation in a moment, quite a bit actually in today's talk. So here we are, saying as a scientific and bioethics community that you can't just stop one and concentrate on the other. But it still led to quite a bit of great interest. And I remember in the earlier days for the International Society, we never had pharmaceutical companies and industry present at the meetings because human embryonic stem cells were just way too hot of an issue. Their shareholders and their board members didn't really want to get involved in that field. But as soon as human iPS cells arrived on the scene, they were suddenly there at the meetings in the front row. Johnson & Johnson, Pfizer, all these companies were there. And they were really interested in this application, which is getting control healthy, so to speak, healthy iPS cells from healthy donors and comparing the development of those into various cell types compared to disease-specific or patient-specific iPS cells, and kind of first seeing what are the differences in development to sort of unlock the black box of human development, so to speak, in how these genetic diseases unfold, and also to get a limitless supply of these disease cell types to do massive drug screening tests. And there's quite a bit of interest in industry to use these cell types for speeding up the drug discovery process. What was really interesting at that time was this kind of activity of using existing stem cell lines to then differentiate various cell types and do your work in vitro. So everything is being done in the dish. You're not going to, at this point, have plans to transfer any of these cells into an animal or to a human being. It's just all done at the lab bench. That type of in vitro research was considered to be the least controversial, the least kind of to worry about. And in fact, in all the guidelines that the International Society drafted, and also California, the National Academy of Sciences, all of them, we've all said this is just kind of an administrative review function. Like, we just need to know, is there ethical provenance of how you got the informed consent for the cell line? And are you just, taking care of like lab safety protocols? And it was really just, no voting. It wasn't like anybody in the committee needed to debate any of these protocols and vote to approve them. It was just keep the administrative office informed of what was happening. So it was thought to be very, very low bar, uncontroversial. What's interesting to me now is that this is part of that answer of like, just you wait, hang on to see what the controversies are with iPS cells. Now what I'm finding is probably the most controversial areas have to do with in vitro research. It's not going into a human or an animal yet, a lot of it. And it's all about differentiating or kind of manipulating these pluripotent stem cells to do certain kinds of things at the bench side, making models of disease, et cetera. There are certain forms of that are actually becoming the most controversial, okay? So I think that's kind of a funny irony there. So the first indication that this was something to keep an eye on was this 2014 paper in Nature Methods. It's called the Warm Flash paper. I love that title. It's because Area Warm Flash is the first author on this. And what they found was if you take any pluripotent stem cell line, this is any embryonic stem cell line, human, or induced pluripotent stem cell line, if you closely pack them together in a micro-pattern dish in 2 dimensions that arranges them in a particular circle of a particular diameter, time and time again, if you just give them just a little nudge of BMP4, a little bone growth factor, they self-organize into this pattern, and it happens over just a couple of days. This again and again, this is remarkable. So the self-organizing capacity of these identical, rather generic pluripotent stem cells. What happened? Well, you got the outer layer that formed what looked like endoderm lineage cells. Next ring in is mesoderm, and then you got ectoderm. And it happened again and again and again. It's something about the outer ring not having cells on the other side. There's some kind of signaling process that kind of cascades toward the center, and you get this pattern embryo-like disc. Now it's not very accurate because it's flat and it's two-dimensional, right? But this was very exciting, and it kind of showed, well, maybe there's something to it. Like when you put the cells together, they self-organize, they talk together, and they do these remarkable things. That's rapidly gone into 3 dimensions. And so we have, examples of like mouse embryo modeling on the left. We have the human cases. There's just been, report after report. And there's been really interesting recent research where we can make distinctions between embryo models that self-organize and represent pre-implantation stages and embryo models that try to represent post-implantation stages. And then the other distinction between, besides pre and post, is whether they're complete or aspire to be complete models, trying to represent everything that you could possibly try to do, including all the extra embryonic material, and those that are incomplete or what are called non-integrated. It's just looking at one particular part of it. So there's a lot of really interesting activity happening. When this was just starting to take off, so after the warm flash paper and when some of the more 3D work was happening, I thought this is a good time to revisit some of the policies around this area of research. So initially, I submitted a manuscript to Nature. It was solo authored at that time. It got accepted, and it was a commentary about how embryo modeling research is starting to kick up a need to revisit this rule that's been in place for almost 40 years, which is you can't do consecutive days of culture of a natural human embryo beyond 14 days. It's called the 14-day rule, right? So you can get an embryo from a fertility clinic, donated to you to do research, but you can't consecutively culture it in a dish beyond 14 days or the beginning of what's called the primitive streak. And what I was wondering was, especially the post-implantation models, how does that square up against the 14-day rule for natural embryos? And so the whole point of the commentary was sort of revisiting what this rule means for embryo modeling. So it was waiting, it was kind of in the queue for publication. And then a Nature contacts me. I remember this distinctly. I was at my son's track meet, like my cell phone rings, and they said, well, we have 2 other papers that are in the pipeline. There were actually, it was natural embryo cultivation. So there were 2 teams, including the team that had the warm flash paper, which was the flattened embryo. There was a team of Rockefeller, Ollie Bermelou's lab, and Magdalena Czerniszka-Gietz's lab at that time in Cambridge, UK, that took fertility clinic embryos and took them to day 13. And they stopped at day 13 in the UK because by law, you have to stop before day 14. And in New York, it was just by guidelines. So they voluntarily stopped there. They said, this other paper, 2 papers are coming out. Could you modify the commentary and kind of add that bit about like the new embryo research is happening? So I added a couple of co-authors, and we said, we need to start talking about this issue. So this was 2016, right? And there's nothing really happened. Nobody was meeting to talk about this, but the research kept proceeding along. So a few years later, in 2018, some of the embryo modelers and I, including Magdalena and others, we decided, well, look, we need to start debating and having discussions about this. So we keep like publishing like the call for meetings and discussion, and nothing really happens. It was getting frustrating. Meanwhile, the science is just clicking along very quickly. So soon after that, this paper came out. And I had the opportunity to work with the PI of this group. This is Jingping Fu at the University of Michigan. And Jingping is trained as an engineer. And he was frustrated at the ways in which the embryo models, the earlier stuff, this stuff, was all very kind of hit or miss. In one culture system, you would have different sizes. They would develop at different rates. They weren't very uniform. So he didn't like that because he thought experimentally, that's not a great system. You really want scalability, reproducibility, controllability. So as an engineer, he developed this system. It's a microfluidic, very simple 2-channel system. You load the pluripotent stem cells at the top of the system. You tilt it 90 degrees, and the cells kind of migrate through these channels. They get hit with this, chemical bath that then initiates embryo modeling formation. So in just a couple of days, you get what looked like an incomplete model, but incomplete of maybe a 10-day-old after post-fertilization embryo. You get that in just a couple of days. So he developed a system where you can generate at will hundreds, very large numbers of embryo models for all kinds of scientific uses. So what would be some of the things you do with this? One would be to screen drugs, to see whether some of the drugs that women take while they're pregnant to see what effects they may have on various aspects of embryo development, for which there was really no data at that time and still today about what might happen. So it could be very useful for drug screening. Another kind of side effect that they didn't anticipate but was very fortuitous was at the outer edge of these, just after a couple of days, you get the migration of what are called primordial germ cells. These are cells that eventually might, under the right conditions, become sperm or egg. And so there are a lot of researchers who are very interested in the kind of like fertility research. And they contacted them right away because they said, this is actually could be used as a primordial germ cell generator. You just get like any patient or someone who has fertility issues, make iPS cells, load them into this, and then you can just generate as many PGCs as you want for research. And so there are all kinds of really interesting applications of this. But I asked them, did it have to look like this? Because it kind of looks like an embryo printing system, right? I mean, you can just make hundreds. And he said, well, yeah, I mean, it kind of has to look like that because if you try to make your embryo models one at a time, it's kind of like baking a cookie one at a time. They're not going to be very uniform. But if you put them all in one batch under the same conditions, they're going to be much more uniform. And that's what you want for kind of the drug screening and the different uses. By the way, besides drug screening, there's another important aspiration for this kind of work, which is to try to develop them to the point where you can get some clinically relevant cell types and tissue types for possible transplantation into patients, kind of like not just the primordial germ cell generator, but just maybe like hematopoietic cells or other kinds of cells very early on. Let nature kind of create the cell types of high quality that you're going to have a very hard time scaling up and making in the lab, one by one. If you can put them into this system and kind of generate possibly clinically relevant cells, that's another really important aspiration. So hence the desire to create very large numbers at scale. Well, here's what I told Jinping. I said, look, you are in the state of Michigan at Ann Arbor, and cloning, going back to the embryo wars, cloning is illegal. And it's very not well defined in the state of Michigan. So I said, for every one of these is one count of human cloning in your lab. So I said, please, I don't want you to go to jail. Please make sure that they're not complete, or nobody could reasonably argue that they might be a complete embryo or might be possibly viable under the right conditions. Try to always leave something out. So that's what they've been doing. So in that paper, in their methods section, they made this statement. They're incomplete. Can't form yolk sac, placenta, and they can't do lots of things. So therefore, they're not real embryos. They're embryo-like, they're models. And I think that that's a good way to go if you can just answer your research question by making models that kind of just do what you want them to do, but nobody could confuse as being functionally the real thing. Then I think you're probably in safe ground. At least in Michigan, you're in safe ground there. But of course, not everybody listens to that, right? To that kind of advice. And now we have people who are really charged up about this kind of work, which is again, pre-implantation and complete as possible. So what they've done, this is Jun Wu's group at UT Southwestern and Jose Polo's group in Melbourne, Australia. What they've done is they've tried to recreate what are called blastocysts, which is the stage in which you get human embryonic stem cells from a fertility clinic. They made embryo models of the blastocysts, and they call them blastoids, or Jose Polo called them iBlastoids. I don't know if he's got an Apple connection there or something, but essentially two different methods to get to the same result. The top column is Jun Wu's method, and the bottom one is Jose Polo's. Jun Wu, you can either use human embryonic stem cells or you can use adult human cells that are reprogrammed into iPS cells. Get them to make in just a few days the blastoid. Similar approach with Jose Polo, you end up with kind of this blastoid thing that kind of looks and in many ways to the fertilized egg here, where you get the human embryonic stem cells. I asked Jun Wu, can you actually get human cells like skin cells, blood cells, make them into pluripotent cells and get a blastoid? And then from this, can you get embryonic-like stem cells? He said, yes, you can. And then I kind of wonder, well, what do you call those? Because it's really not really an embryo, and it's not an iPS cell. It's kind of like from skin to iPS to then blastoid to then embryonic-like stem cells. Really quite fascinating. The other thing that I thought was really interesting about either of these methods, but especially this one because they've used human embryonic stem cells, is for human embryonic stem cell derivation, you get this from the fertility clinic. This is about day 4 or 5 after fertilization. And you open up what's called the trophectoderm, the outer layer, and you take out the inner cell mass, and then you get your human embryonic stem cell line. we still have, human embryonic stem cell lines that were derived in 1998. You can take those and reconstitute the blastoid, which seems to be very close to this. Right, so think about that for a minute. You can rescue the original blastocyst. You can make tons of copies of that. So it's a really interesting, kind of that theme of what's old is new again. You can kind of take the old stem cell lines and reconstitute this. It's really fascinating. These don't seem to be developmentally competent at this point, not yet, but it's very early days. There are, some spatial organization issues in the inside. And all the monkey ones that they've made from monkey iPS cells, they tried implanting those. You get a pregnancy response for about 10 days, but they don't go very far in the womb. What is interesting though is Jun Wu and others who do this work with the human blastoids, they're actually interested in using this technology with animal cells to rescue endangered species. Right, so this would be safer than cloning, which is nuclear transfer. Right, the Dolly experiment where you get an egg, unfertilized egg, you take out the nucleus, put it in the nucleus of a somatic cell. Well, in that case, it can sometimes lead to some birth defects and some health problems. And it also has different mitochondria because you're using the mitochondria from the egg donor. In this case, it's more purely a clone because it has the same mitochondria from this original cell line that you started with. So on the one hand, they're trying not to use this for human reproduction and trying to say there's a bright line between kind of like what we want to do with embryo modeling and wrongful uses for human reproduction. But at the same time, these teams are also trying to use it for animal species rescue. So at some point, I hope, succeed with the animal models. And then there's a question of whether or not that might affect our attitudes and policy around the human blastoid work. They're getting much more complex now. This is some of the post-implantation modeling work. These are both mouse models. One of them is a real mouse embryo, and the other one is a mouse embryo model. The one on the bottom is the model. They're getting pretty close. This is Magdalena's work. So the work is going along pretty quickly. The international guidelines that I helped draft, the latest version that came out in 2021, there are certain things that we've tried to do to address this burgeoning area. I will say these guidelines are trying to do a few things. One is we're just trying to get through the next 5 years, folks, okay? We're not trying to come up with the guidelines for all of eternity from this point on. It's just what do we need to do to kind of let the science show us what it's capable of doing in a responsible way in the next 5 years. And then we have to reassess where are we to then come up with new guidelines. So at this point, back in 2021, we at least made the distinction between the complete and incomplete, the integrated and non-integrated. Non-integrated, we kind of think you don't really need to worry too much about that because it's really they're not going to go anywhere beyond just what you're studying. But it's the integrated ones that are going to be a little bit more, ethically charged. But regardless, there should be no transfer into a human or non-human uterus. These embryo models. I said that, we have to kind of revise these every 5 years or so. My recommendation right now to the committee, if we were to form again today, is we have to be clear about what we mean by human or non-human uterus. Because one of the things that has to happen now to move the embryo modeling forward is cultivation beyond the implantation stage. And to do that, you have to create a womb-like environment in the lab. Right. So again, this is all in vitro. It's not going into a real animal. It's not going into a human, but you have to bioengineer kind of like the support structures around it. And sometimes you might have to use human cells, like uterine cells. You might have to source them from clinics and cobble together what kind of might be functionally, at least for that little developmental period, a uterine lining and embed your embryo model into that. So let's be a little bit clearer about what we mean by a human or non-human uterus. I'll tell you, when we formed these guidelines, we had in mind a whole animal or whole human being and the natural uterus there, that environment. But I think we need to really think through kind of, might we also mean some of these artificial systems? Because that's really kind of, I think, where the next step is going to be in this research. The other thing to note that we know we're very much aware of here is the legal definition of the human embryo has shifted over time. Now it's less about how you made it and much more about what it will become or what it could possibly do in the future, right? So if you look at the NIH definition and the definition kind of related to federal funding, they define the human embryo as any product of fertilization, cloning, parthenogenesis, or a combination of human diploid cells. That pretty much covers it, right? That covers the embryo modeling, the integrated stuff. It's really about what will happen if you transfer this into the womb. So what I'm worried about is those embryo modelers who are saying, no, don't worry, we're not going to transfer this into the uterus of human or non-human, none of the human embryo models, but we're going to work very hard to work on non-human primate and other, white rhino, other endangered species and extinct species. As soon as they start getting live births that way, right? What's old is new again. You're going to get the whole Dolly experience again. So in the Dolly experiment, right, as soon as they started making sheep and then cats and cows and pigs and other animals, even if they never transferred the human cloned embryos into a uterus, people looked at that data. They said, look, it's making all these animals. You got a whole barnyard full of cloned species, right? And that really led to, for example, the Michigan law about making any kind of human cloning illegal in the state of Michigan, like a big backlash against that kind of technology using human cells. So you actually don't have to have somebody kind of break the rules and transfer into a uterus and a human embryo model to test whether or not they're viable. People will look at the animal data, the regulators and policymakers and lawmakers will look at the animal data and say, well, that's good enough. That's all we need to then turn the screws down on human embryo modeling. So I'm really curious to see where things shake out in the next few years. So the animal work is going to be really very important for kind of the future of the human work, and also in the human work, the support systems that are being created, the womb-like environments. Those are also two big areas to look at. Okay. So, again, I mean, still no movement on having any kind of like national or regional discourse around the 14-day limit. So we were getting a little bit frustrated because earlier in this version, we relaxed the 14-day rule. We said, actually, in principle, it might be possible to go beyond 14 days if in your jurisdiction, it's not illegal to do so. And you have a lot of public dialogue. And those are pretty low bars actually to have. So we needed a little bit more guidance on like, what does it mean responsibly to go beyond 14 days? So we came up with this article. I will tell you on this one, where we tried to sketch out some reasonable approach of what would it look like to kind of like explore just a little bit beyond day 14, like what would those kind of protocols look like? What would the standards look like? We got, this is a very interesting experience of science for a couple of reasons. One was it went through 8 reviewers, which is kind of a lot for commentary. Usually it's like 2 or 3, it's 8. And all 8 gave it the thumbs up, so we felt good about that, okay? But then when the editor sent it back to us, they asked me, could I explain a little bit more of the history and the context of the 14-day rule? Because the editor didn't think that the science readership would understand that background. So think about that for a second, because so many people have said to me, well, the reason why the 14-day rule is so important internationally is because it comforts the public. It gives them assurance the scientists won't go too far. And I thought, but the public has to know about something like that to be assured by it, right? They have to know something about that. And I thought if the science readership kind of needs further information, I'm not really sure my Uber driver will kind of like know what the 14-day rule is and feel comforted by that. So I think that was a really interesting kind of dynamic to witness there. So anyway, so we've sketched out kind of what responsible innovation looks like there. Now, it should not be surprising to people in the field that stem cells have this power to recapitulate human development of the human embryo, because we've known that they can do that for different organ systems. Right? We've known for almost about a decade now that you can put stem cells in culture and they can recapitulate different organ systems called organoids that are 3-dimensional and just very rudimentary. So we have brain organoid, airway, we have liver, different examples of this. Now, the next step that I think is really interesting about this is assembling them together, right? So my colleague Sergio Pashka at Stanford, they've been doing some really interesting work where they assemble different organoids together. So for example, one brain organoid is not able to recapitulate every aspect of the brain, all the regions, but you can put some of them together for brain hindbrain. You can co-culture, you can put them and create what's called an assemblage or an assembly of different models. Sergio even went so far as to make a combination of brain organoid, spinal cord organoid, and muscle organoid, put them all together, and you got signals from the brain organoid going through the spinal organoid and twitching the muscle organoid. So there's some like really interesting ideas of maybe combining organoids together and do like drug screening or kind of seeing how these systems interact. And all the concern seems to be around whether or not these kinds of entities will create any kind of conscious experience. And so because of that kind of concern, we did this project which was me and the PIs of these three labs, Sergio, Paula, and George. And in over 2.5 years, we were told by the NIH Brain Initiative to kind of try to identify what are like the main issues that we need to get through in the next 5 years, right? What we said right away was there's not much to worry about for the consciousness issue because these are just little brain organoids in a dish and they're not connected up to any kind of sensory input output system. They're missing some really key cell types like microglia. You would have to add them, you know, artificially. And so really the main issues that we found, the more near term, was validation. How do you know that these brain organoids or other organoids will actually recapitulate nature? I told you that there was a lot of interest in industry of using organoids and other differentiated tissues for drug screening. The number one question that these researchers get from industry is, these are pretty neat, these are cool. How do we know that this is actually accurate of the human condition? What do you have to do? Well, these grow and go through fetal stages. You have to compare it to primary fetal tissue. Validated. That's a really red, hot button issue. So validation comes up again. How do you know that the model you're creating is accurate? The embryo models. I told you they're trying to do the post-implantation models. So how do you know that these embryo models are accurate? You have to validate them against embryos that are from fertility clinics that you've grown past 14 days. Right? Everybody would love the day in which we can use all kinds of embryo models for drug screening. We can make hundreds of them. They can maybe create some clinically relevant tissue. But until you really know that that's actually an accurate representation of nature, then industry is not going to be all that interested, right, in for drug screening or for anything else. So you have to compare it to the real thing, and to do that, you have to go past 14 days. So there's going to be a lot of pressure. So initially, when I wrote the first commentary for Nature about like, oh, the embryo modeling kind of raises this policy question of should they be kept to 14 days or the equivalent or less, there's going to be so much more pressure on that question when you get people who really want to take it to the level of commercialization and validation. You have to compare it to the real thing. So there's a lot coming down the pipeline. So we ended up publishing the results of this brainstorm project in this issue of Nature Neuroscience, and we explain like what are the near-term issues. It's going to be validation. It's going to be better science communication of the teams, and it's not the consciousness issue. That's kind of a red herring. Okay. So I'm going to start wrapping things up now. So I told you the big areas of controversy in the old debates were about the moral status of things you're creating in the lab. So that's kind of coming back around again, with iPS cells leading the way. I mean, that's really kind of the main cell type used for a lot of this work because you want to make it patient-specific and disease-specific. And when you get stem cell lines from an IVF clinic embryo, you can't control for the genetic makeup of that at all, right? So iPS cells are leading the modeling work, and the modeling work is kind of starting to cycle back into early life. The whole other issue that I didn't get into, which is in vitro gametogenesis, which is creating sperm and eggs from cell lines, that also, if that progresses further beyond the mouse model, progresses to human, we'll have to start doing fertilization tests of the in vitro derived gamete with the natural counterpart. So there's going to be a lot of, again, returning back to the embryo question in a very unexpected way. So it only took, 15 years or so, but here we are again, back at the old debates. So one of the other really hot areas early on in the stem cell debate was chimera research, so transferring human cells into the animal model to create human models of disease in the animal, and especially neurological chimeras. That cooled down quite a bit when it's found that like the human cells don't survive long in a evolutionarily distant model like the mouse or even the pig. They don't survive very long. So people kind of thought, well, maybe we don't need to worry too much right now about chimeras until organoid transplantation started happening. So the very big worry was transferring human brain cells right from stem cells into laboratory animals and getting integration into the brain. Well, the cells don't survive long enough to really get much integration. They don't do much. But if you take a brain organoid and put the whole thing in, then you kind of get in one step, a very relatively complex human brain structure put into an animal model. And so this is Sergio's work. They transfer human brain organoids into the rat, into a neonatal rat. They ended up getting they put them into one hemisphere at a time, all right? And it ended up growing to take up about a third of the hemisphere over several months. So the human brain organoids were very well tolerated in the neonatal rat and integrated well. And it got to the point where they discovered three really key things. One is there are things that happen to the brain cells in the rat model that you don't see in the dish. There's something about that dynamic environment that makes them grow more complex and show features that you didn't observe in the dish. So they did the organoid culture in parallel. Dish only transplantation, they recover the cells and they said, this is so much more complex. It might uncover lots of other information that we wouldn't get in the dish. That's step one. Number two, they found that it actually integrated into the functioning, the brain functioning of the rat. One was they could see the human neurons in the brain organoid light up when they put it into this motor sensory cortical area that's associated with the whiskers. And when they kind of like stimulated the whiskers, they saw that part of the brain organ, the human part, light up. And then the other one was that they were able to optogenically control some of the human brain organoids by training the rat. Every time it took a drink of water, they would stimulate optogenically those human neurons to the point where all you had to do was stimulate the human neurons and it would go after the water. So it showed like how you can get like a behavioral readout. And that was really significant because it showed they actually integrate, they tolerate the graft very well. And you can really kind of do some like, see if like there are changes in the various cell types you put in that may have an effect on behavior. So exciting for their lab, but it was very worrying for them as well. So when they were about to get this paper published in Nature, Sergio contacted me and said, hey, Insoo, I'm worried about what's going to happen when this paper comes out in Nature. And I said, well, why don't you come down to the Museum of Science and I can interview you and we can talk about your work and release it on the same day that the embargo lifts on your paper. So he came down. It's on our YouTube channel for the Museum of Science. You can see this interview. It's a lot of fun. But we had time to, we talked for 3 hours. We aired 40 minutes. So there's a lot of unused footage, but it was really interesting to kind of be in a position to help translate to the public without him having to go through a reporter and having the reporter kind of clickbait it, reframe it, kind of like allow him and I to just have a conversation, talk about how did you do the research? Why did you do it? What are the concerns you have and what good can it do? Kind of putting all that context into it and the history of why did you have to do this? Why couldn't you do the old ways, right? And so it was for me, a very important thing to be able to try to do for the field. So you can check that out if you want. So all these old questions are being renewed about not only the moral status of, let's say, the embryo models, but also when does personhood arrive, let's say in the, or human-like consciousness arrive, either in the brain organoid in the dish, I would say never, or in an animal model. What's really interesting about the rat model that they did was there didn't seem to be any other behavioral changes or deficits. The big thing was they were worried of possible like bad seizures in the rats because it was a large graft. And they were really worried that the animal would take a major dip in welfare. Actually, you couldn't tell in terms of just like external behavior, except for like the little whisker test and the water behavior test. Acted no different, and it tolerated it very well. And they got all the information they needed using the rat model. I asked them, would you ever want to go to a more complex animal model? And I said, no, at this point, everything we need to know, we can use the rat model and it's doing really well. And rats are a lot less expensive and they're easier to work with. He's not sure if other people might take that kind of work and use non-human primate or use other kind of more controversial species. So this is kind of a wait and see approach on that. But these questions about human-like consciousness emerging, like that came up in his work. It's not human-like at all. It still behaves just like a rat. But we were really at the stage of kind of wait and see what might happen. So where do we go from here? I don't want to keep repeating the old debates again and again. Like in the past, people have tried to address these kinds of questions with philosophical argument and ethical analysis, and that's all important. I love that stuff. That's where I come from. But it doesn't really do much to kind of, I think, move the conversation forward in a productive way. All you really see is there's quite a bit of disagreement, and there continues to be disagreement and disagreement. It's just a given. People are going to disagree. So I think the more fruitful approach is to reframe these issues, not as like moral status issues and, is it right or wrong to treat an animal that way? I think as long as like other institutional and animal care protocols are being taken care of, the real debate has to do with in a society where people disagree very deeply about things like the moral status of the embryo or what human consciousness is, for which there's no like clear scientific answers, it's more philosophical. The real question is, how do you mediate that kind of deep disagreement in a society where science has to have some answer, whether they can do something or not? It's more of a political philosophy question. Because if we keep sticking to the ethical analyses, philosophical arguments, religious arguments, we're going to keep going in a circle again and again. Old becomes new and it just gets recycled. And I really like to break beyond that and just say, is there another way forward? Because no matter what you may think about the moral status of the human embryo or not, around the world, there are very different cultural attitudes about this kind of work. And no matter what, somewhere in the world, it's going to go forward. My favorite quote from Marcel Proust is, "The dogs bark, but the caravan moves on." Dogs are going to keep barking, but what are you going to do with the fact that it's going to keep marching forward? And I would much rather spend time thinking about kind of best ways to kind of reduce harm, and not so much about which cultural tradition, which religious doctrine is the right one. I think that's really going to get you nowhere. So with that, thank you so much for your attention. These are my funding sources, and I'm happy to take any questions. We have about 7 minutes. I wish we had more, but.
Before we take a few questions, I just would like to officially present our Women Lectureship memento for all of your recognition and achievements, contributions in the field. Thank you so much for coming to talk to us today.
Wow. Thank you so much. I already gave my speech, I guess. I want to say
One round of applause, please, for the Vaughn family who makes this lectureship possible. Thank you so much.
Some of the ethics of the consent of the person who donates the cell.
Yeah, that's a really great question. The question was, what about the ethics of the consent of the original cell line donor? That's a really complex issue. So at the international society level, we've wrestled with that. It seems like based on empirical studies of patient attitudes about some of this work, that there's some pretty reliable data that people would want to know things like, are you going to use my immortal cell line, my iPS cell line, for some of this work like embryo modeling and then vitro gametogenesis, maybe even the brain organoid work, like those three hot button areas. And so there's enough evidence to think that people probably want to know. And so the best we could do is in the consent process, say that this is one possible use of the cell line. The problem, though, is most of the iPS cells used for this kind of work have not had any kind of explicit consent for iPS cell derivation and use. Mainly, they were like tissues that were commercially purchased that are disease specific, and people just made them into iPS cells, or they're clinical discards that are anonymized from hospitals, and they just use them. So much of the work that I showed, I would say like 99% of it, most of it, almost all of it, has been without any explicit consent for that specific activity. And so the question is, what's the best way forward? I mean, I guess we got to grandfather those in. And the ethics behind this is always about, sort of respecting the values and the intentions of the original donor, beyond just simply like donating your biomaterials to do good for science, but just a little bit more specifically, because these are very highly ethically and culturally sensitive tissues. So it's usually, so brains and gonads are all what people care about. So if it's like, sac cells you're making from the sample, or brain tissue, or even like, early embryonic like models, people usually want some idea of this is a possibility and they want to opt in or out of that. So there's no real great answer, right? So one is, it's already been done. That's the really well-characterized data we already have. And then going forward, I think the recommendation is going to be, going forward though, you really should, if you're going to make these very controversial kinds of things, you really should get explicit consent, but it's challenging. The other thing is, like, you have iPS cell lines, you don't know if another research team is going to do that, so there's an initial collection, and you may have said, well, we're not going to do that, but somebody else might in the future. So I don't really know what the best solution is. You can't overpromise. You can't say, well, we're never going to do something like that with your cell line, because you can't control what everybody does internationally with that cell line. You can't say that to the donor, because if you say that to them and they say, oh, I'm reassured by what you're saying, I'm going to then participate. If you can't guarantee that you're going to make good on that promise, and you're actually not getting full informed consent there. So we're continually talking about that donor issue. That's a great question. I wish I had an easy answer.
So you talked a lot about the relationship of how technology drives a lot of these questions, principles, guidelines. Is it possible to make guidelines that can stand the test of time of technological progress?
Yeah, probably not. I mean, you can do some general things, right? So you can say things like the guidelines are that there always has to be, review by a committee. I mean, I know that's not a great answer, but at least assure that there has to be a process for continual reassessment as the science progresses. What I would think that we need to start doing now is, because there's so much bioengineering that's being involved, that we probably need some other like framework for bioengineering ethics and ethical considerations in that kind of partnership. But yeah, I think that that's something we have to take up in the next version of the guidelines. But in answer to your question, other than to say, here are the very general ethical principles about respecting people's values and always making sure that the research is aimed at some sort of social good. Other very general things like there should be a very clear scientific rationale and lack of alternative methodology to do something very controversial. We're actually working on that paper right now. With the exception of those kinds of very general guideposts, it's hard to be very specific because it's moving so quickly. Except just to say, like, these are the kinds of value commitments that we should uphold. How do you interpret that in your own research locale? That's going to be variable as well, right? Science is international, but it's conducted locally, and it's hard to find that right balance.
Earlier in your presentation, you mentioned that there are structural and organizational differences between the blastocysts and the blastoids.
Yeah.
Does that have a negative impact on research in terms of validation?
It depends on what you want. So the question is, like, the blastoids and the blastocysts, they seem to be a little bit different. Do those differences matter? From what I'm told, it depends on what you want to do with the blastoids. So I think if you want to use them for reproduction, so this is the animal case, I think it is a big issue, a big stumbling block, because they, I mean, just experimentally, when you put them into the uterus, like, they don't really go anywhere. So there must be some major hangup. If you wanted to use the blastoids to study some other aspect of that stage of development, it depends. I mean, there are some unknown cell types. They don't really know what the heck are some of these cells in here. They don't know exactly, what they're doing and what effect they have. And then the spatial organization is a little bit off. But if you want to just kind of show that it's possible, actually, one interesting area is to create blastoids that are very genetically specific. Right? So you can't breed human embryos and get like genetically specific, right, embryonic stem cells, but you can get iPS cells that are like have a very specific genetic profile, or do genetic altering of your cell line, and then you make a blastoid. So there's kind of some really interesting developmental work to kind of see the role of genes. And so it kind of really depends on what your research question is, whether those differences are deleterious or not. Yes.
Because I'm thinking about how to word this question. So I guess hanging along with what you were floating your remarks were, if the caravan's going to move on, I guess why bother discussing?
Yeah. So if the caravan's going to move on, why bother discussing it? Because it can careen off in pretty bad ways. It could leave people behind socially that are not being like benefited from the research, or lots of things that could go wrong. Right. So on the one hand, people have told me, well, Dr. Hyun, you're being pretty fatalistic about all this. And I don't know, I like to say realistic, because you can't kind of stop it from chugging along, but you can kind of change who's part of the discussion and kind of where the priorities are. Right. Yeah, I just think it's just a matter of time, honestly, before ex vivo gestation is a possibility, because you have people who are working on pre-me support, are trying to push down the time of viability, and you have the embryo modelers trying to like drive up development, and eventually that's going to need. And so you just got to be prepared for stuff like that. So I think that's why we need to be involved in the discussion, because I think the technology is coming. It's just kind of a matter of limiting the bad that can happen and maximizing the good. Oh, sure. Let me see. Oh, right, nomenclature. Yeah, so that's a big one. So Sergio and others have kind of proposed like not using the term brain organoid or cerebral organoid, definitely not mini brains, right? They suggested neural organoids. I think it's a little, that to me is a little too general because that would include the spinal cord organoids. But that's where, but it is an effort to kind of like reframe the labeling or the discussion of what you're calling these things to kind of then help minimize some of the heat. So another good example is people kept calling the embryo models synthetic embryos. So there's actually a paper coming out pretty soon, or maybe it just came out. Maybe it just came out in Cell Stem Cell about like why you shouldn't call them synthetic embryos. They should just be called embryo models. And I think it plays a very important part. And definitely the scientific community and the international society is very mindful of what we're calling these things. People wanted to try to call human research cloning SCNT because that just kind of sounds kind of sciencey and people don't really even know what you're talking about when you see SCNT, somatic cell nuclear transfer. But I think there's a limit to kind of the terminology because I think some people are just they're gonna accuse you of trying to shrug off the debate by just calling it something else. I mean, that happened in the very early days when the 14-day rule was established in the UK. Anne McLaren, the great developmental biologist, argued, well, really before like implantation and all that, it's really a pre-embryo. It's not, they don't really consider it developmentally an embryo because it could split, it could do all kinds of things. And so she proposed pre-embryo, but immediately the committee said, no, people are going to accuse us of sidestepping the ethical issues, the difficult questions by just relabeling it a pre-embryo and not calling it a real embryo. Now there's a whole debate about whether we should change the legal definition of a human embryo to kind of better accommodate the embryo models. But the human embryo redefinition, so this is some of my co-authors on the embryo modeling paper, they also regrouped and without me, they published another paper where they argued that you need to reframe the definition of a real human embryo to include all the extra embryonic material. I thought that could be kind of confusing for policy and for fertility clinic use because people call the embryos in the fertility clinic, the fertilized eggs, they call them embryos. They talk about embryo transfer, embryo storage, and it doesn't have all the support tissue around it, not yet, right? So I think we just need a better definition of what an embryo model is instead of coming up with a better definition of the human embryo, the natural human embryo, to then show how far away the embryo models are. Let's just be clear about what it is that we're talking about, embryo models. But yeah, but many people have pointed out, be careful about what you call them. And that's, again, I think a strategy that's useful up to a point, but I think opponents are going to view that as just a workaround, a step around.
All right, I think we're about out of time. We have to get into your final appointment for the day. But join me once again in thanking Dr. Hume for a great time.
Okay.